Resume recording. Gotcha. Okay. All right. Okay. Perfect. Thank you. Yeah. Okay. So any questions before we start, like from yesterday's and the past few days lectures? Well, I know there's too much things are going on for all of you, so take your time, as you sink in those lectures during the weekend, I'm sure they will have a lot of questions, and feel free to, email me, or we can meet as a group, like I said. And then tomorrow, I think we will have some leftover lectures from today that I would like to go over quickly in the first half, or in 1 hour, we'll go maybe in the first 15-20 minutes, and then will ask me questions, or I'll just kind of give you an idea of the exam format for my sections. Okay. And it should be pretty straightforward if you just follow my lecture notes, and then we'll just indicate some of the priority things that you would like to focus on in busy slides. Okay. All right, let's then get started. And I think if we left here yesterday, if I'm assuming that nobody has any questions, at least today, so we will move forward. Okay. All right. Okay. So today, and then all of today and then tomorrow, we are going to talk about how best we can integrate our concept and the knowledge that we have acquired until now. That means we are going to how things add up. Okay. So we are going to utilize our knowledge of tumor suppressors, oncogenes, tumor modifiers, and then we'll try to put it in the perspective of a whole full circle. Okay. So that means we are going to study cell signaling, and the cell signaling, as you will see, will make things much more clear in the context of everything, or most of the things that we have learned, how we can put that in a context of step-by-step fashion. The two other things that we are going to go over today is a multi-stage process of carcinogenesis. And the multi-stage process of carcinogenesis is important because, keeping in mind that it's a longitudinal process, whether in the terms of the preventions or therapeutic intervention, or as of treatment, whatever modalities you are going to focus on moving forward in the future. Because of being a multi-stage process, it's also interesting to appreciate that there is a lots of scope for interventions at any particular stage. Okay, so that's the second thing. And then the third thing that we are going to go over today, most of the today, and I think probably a little bit tomorrow, is to identify what are the hallmarks of cancer. Okay, so that means what are the main characteristic features that I, as a cancer cell, need to acquire, or what are the hats that I need to wear in order to qualify myself to be a cancer cells, and what are the different attributes I might have as a cancer cells that can help me stand out in comparison to other normal cells, or in the cells which are in the intermediate stage. Okay, so this is an overview of what we are going to go over today and the rest of the tomorrow. Okay. So, so that, as you see, why it is important, we know now by this time that it's important to study cancer cell signaling. And the reason behind is, it's all just helps us to understand, put things together, how we can better understand the mechanism of actions of any drugs. Eventually, that is our main goal. But identify which targets are deregulated, which are the major, which specific molecules should we target, what are the key areas of focus, so on and so forth. Okay. Now, this is again a simplistic, this is a slide that I often use, or I always use, to enforce the concept that during this process of our understanding, what we have always felt initially, that it is a more of a parallel process, deterministic pathway, where each tumor would follow an individual pathway or a signaling, and then that would lead to eventually its transformation into cancerous stage, right? Now, that's probably, and it's never the case, because it's more of a plastic pathway, or what it means, there is a lot of plasticity, lot of flexibility for each tumor to choose any pathway it wants, and it's more of a maze where individual tumors of a given organ type, tissue type, let's say, can follow multiple mediators or mode of actions, and ultimately leading to the same goal of transformations. Now, during this process of transformations, there are a lot of changes that happens, and there are phenotypic changes, mutational changes, and I added epi mutation, which means changes that is happening at the epigenetics level, right? That you are kind of already aware of by this time. So that's our starting point in mind, but nothing to, so just to keep in mind, there is a deterministic pathway and there is a plastic pathway, and that's the more accepted pathway is the plastic pathway, which is a lot of more flexibility given to the tumor cells to reach their final destinations of getting transformed into a cancerous cell. Okay. So we are going to address some of the major questions that would come in our mind. And these are, what are the signal transduction pathways involved, or at least the key signal pathways? What are their cell biological outputs? That means how these signal transductions will be at all important for me to know, so that it can impair or impact some of the cellular outcomes such as transformation. Then always it hones our mind that what are the targets of these pathways, right? Means, these targets, why do we care about? And ultimately, all we need to know is that what are their ultimate goal of this pathway, so that we can find out a way to intervene. And then also how these targets introduce changes leading to cancer. That means how they eventually contribute in the process of cellular transformations. And last but not the least, of course, we already know that how best we can modulate these pathways for our benefits, to human mankind. Okay, so these are our major focus of attractions. Now, in this slide, this is really like a top view. This is a 30,000 feet view of what is going on in terms of integrating this process or signal transductions. So in a very simplistic way, if I'm a cancer cell sitting here, and then there are lots of factors, lots of environmental factors, growth factors, which are surrounding me as I am exposed, as I walk through the corridor, and these growth factors in the form of acts as a signal, right? Now, these signals or the growth factors are being, received on the surface of these cancer cells or this cell. And once they're received, they are basically, these signals are transduced. And these signals are transduced where? Their signals are transduced eventually to the core, which is the nuclei of the cell or the nucleus of the cell. And this process involves multiple post-translational modifications, not limited to just phosphorylations. But phosphorylation is one of the major key modifications that happens during this process of signal transductions, where eventually a signaling cascade from an extracellular signals goes and transforms and gets transduced intracellularly towards the nucleus, with an ultimate goal that it will allow transcription of genes, help produce more cells into the cell cycle, genes that are involved in the cell cycle progressions, and help in the cellular transformations. Okay, so this is an absolutely, like a big picture, if you would like to say. Okay, now in the next few slides, I'm going to introduce with some of those major growth factors and their receptors. Now, for the sake of your exam purpose, you can just focus on the first two, the PDGF and the EGF. Okay, and now this is again not an exhaustive list of the factors, but these are one of the major factors that often are known to be altered in different cell types in the different cancer types, but predominantly this PDGF, EGF, which is the platelet-derived growth factors, the endothelial growth factor, or epidermal growth factor, and also to a certain extent FGF are the major key players that controls in multiple cell types and control signal transductions, which is mediated through their receptors. So this is a list of all the growth factors, those signals, extracellular signals, which are present. And in the subsequent slides, you will see the corresponding receptors, okay? Now, this is a busy slide, right? But the most important take-home message from here is two. First thing is that, let us see, if you look closely into this slide, you will see that there is a lot of variability extracellular. So if this bar that you see here, down here, this is the cell membrane, which separates from keeps the inside in and the outside out. So then externally, every receptor have a unique domain, structural configurations, okay? So EGF, or epidermal growth factor, this is the corresponding receptor is EGFR. We have PDGF, the corresponding receptor is PDGF receptor, PDGFR, right? But if you look closely, and whenever if you get a time, you will see each one of those receptors have a unique conformations, unique configurations, which makes it such that every individual growth factor will bind and to its own receptor. There is absolutely no scope for the individual factor to bind to a different receptor. However, if you look just underneath, so if you dive deep inside, go inside the cell, the intracellular domains are pretty conserved, okay? You can say, and I'm not going to go into that details, but just to keep in mind that all of these extracellular signals or antennas which are present, they are all unique. They are receiving individual signals from individual growth factors. Upon binding to their individual growth factors, these receptors are producing and transducing signals in a very similar way because of their similarities in the intracellular domain, okay? All right? So that means EGFR will bind to the EGFR receptors and most likely will transduce MAP kinase signaling pathways. And so it would be the same case essentially for PDGF or VEGF receptors. Here, VEGF, vascular endothelial growth factor, will bind to the VEGF receptor. So that's one important thing. The other important thing, by this time you probably have already noticed, or if you have not noticed, that each growth factor, the simplest way to remember is that just add an R at the end. So VEGF, the receptor will be VEGF receptor. That means VEGF-R. There won't never ever be a situations that VEGF would bind to the EGF receptor. So if there is a questions, if you would like to keep that in mind, the growth factor will always bind to its cognate receptor and then helps in the transduction of the signaling from an extracellular domain to an intracellular domain, which is this, all of this extracellular goes to the intracellular domain and converge into multiple signal transduction pathways, ultimately leading to the core nucleus. Okay, does it make sense? All right. So next, we are going to take an example of, let's say, any growth factor, because we have a receptor tyrosine kinase, and as you know, many of these receptors are receptor tyrosine kinase receptor. Like growth EGF will bind to the EGFR, it's a receptor tyrosine kinase. Same thing, VEGF is VEGF receptor tyrosine kinase. So we will, let's say we have some growth factors. In this case, let's pick our favorite one, let's say epidermal growth factor. So in this case, epidermal growth factor or factors, multiple of them will bind to their receptor, okay? Epidermal growth factor receptors. And then their intracellular domain, there are some other proteins which are here. Now, the important thing here is once upon binding, the signal transduction takes place. And you remember our RAS, our favorite RAS protein, okay? So the RAS is now getting activated from a GDP-bound inactive form to a GTP-bound active conformations. Now, upon this switch happening from an inactive to an active conformations, that leads to an activations of the downstream signaling, which is the RAF family kinases, like the BRAF, for example, okay? Which then further activates the downstream, the MEK family kinases, which is the MAP kinase pathways, which is the downstream effectors, goes to the ERK signaling pathways, and then the ERK, which gets phosphorylated and gets translocated into the nuclei of the cell, eventually helps and binds to the c-Jun and the c-Fos transcription factors, which helps in the transcription of genes such as cyclin D1 And we now know by this time that cyclin D or cyclin D, for example, and specifically cyclin D, is involved in cyclin CDK4 complex or cyclin CDK complex and is contributing in the cell signaling and cell cycle progression. Okay. So this is a basically a step-by-step process how multiple signaling or extracellular signaling is intercepted on the surface of the cells, initially normal cells, but then eventually they transform cells, and then the signal transduction takes place. So most of these alterations that is happening from a normal into a benign to a cancerous stage is alterations in each one of these steps or many of these steps or multiple steps that are involved in this process of cellular transformation. Okay. Now, we also know that there is a protein called RAS gap, or the gap which basically helps in switching back the GTP back to its inactive conformations, GDP, until unless there is a mutations in the RAS which helps in the constitutive activations of the RAS signaling pathway. Okay. Now, what is now interesting for you to appreciate that each one of these intermediates of the signal transduction pathways could be mutated in multiple cancer types. For example, and this is just a list of some of the examples that you detect mutation in the KRAS in almost like 20% of the small cell lung carcinomas or over 60% of pancreatic carcinoma. Okay. That being said, if RAS is mutated, you already know that there is uncontrolled cell proliferation. We heard yesterday that if RAS is mutated, then there is activations of the MAP kinase signaling. The RAS remains turned on or stuck in its own position. Thus, constitutively, these pathways are altered. That means you get constitutive overproduction of ERK. ERK then produces excess of the c-Jun c-Fos binding and excess of cyclin D, which definitely is driving much, much more of the transformations. Now, on another side, people often detect that there is a genetic amplification of cyclin D1 genes in cancers such as the breast, colon, and hepatocellular carcinoma, which means independently, one or many of the signal transduction components can be either altered, as in this case, amplifications. And we now know that gene amplification is a process by which these cellular transformations can take place. What is also interesting that this, you see the receptor tyrosine kinase, HER2 receptor tyrosine kinase is overexpressed in almost like 30 to 40% of breast cancer patients. That means, what does that indicate? That means if you have more receptors on the surface, overexpression of these receptors, you'd naturally expect an overproduction of this RAS signaling by default, even though there may not be RAS mutations. So that means there is clearly two things. First thing is that there is a redundancy in the pathway. That means you do not need to have everything mutated all at the same time at the same cells in cancer, all cancer types. Or that's the first lesson, but alterations in one or two or few of those intermediate key structures, key stage, can itself lead to the culminations of cellular transformations. First. The second important thing is that there is a lot of scope of interventions in terms of preventions or therapeutic interventions. That's the good part, right? That means if you are in a company or if you're developing your own drug or screening for new drugs, you have an opportunity to find new developing strategies for, let's say, MEK inhibitions or RAF inhibitions, okay? And that is exactly the case that is happening these days. And I won't go into what Dr. McFell will teach, but I'll just give you a flavor. Let's say there is still up to date, there is no good RAS inhibitors on the market. You look up, nothing that is really effective in preventing RAS mutations. But there are some BRAF mutants, which basically targets the RAF pathway. But then again, for reasons we know now, resistance and alternative pathways, the MEK can get activated. And then there are MEK inhibitors, such as tramitinib, okay? BRAF inhibitors are there, vemurafenib, okay? So just keep in mind that those she will go over with details, but there are multiple opportunities of therapeutic interventions, either alone or in combinations, along with the traditional therapy like radiation therapy or surgical resections. Okay, so it's more than what you see here, but still this actually gives you a global view of what's happening deep down in at the cellular level. Questions? Okay. Again, those who are on the Zoom, please do, post your comments on the chat. And if I see that, I will probably see that at the end of the class, I will try to respond, or shoot me an email so that I can respond to your questions. Okay. And you can also unmute yourself and feel free to interrupt. So moving forward, so this, you will see this slide, and the next slide will give you just a flavor of that. And so, for example, we are talking about a BRAF inhibitor, okay? And so there's, like I said, this BRAF inhibitor will block and prevent RAF activations. That means everything that is upstream, let's say if there is a mutant RAS or there is overproduction of kinase, activity of the receptors, it is expected to be blocked. But in ideal scenario, you need a combination, the combinatorial treatment. So, but that's kind of a classical example to show that if RAF is blocked, you would expect that MEK and the ERK signaling will be stopped, or over excess of the signaling will be mitigated, and then one can normalize. So these are all active areas of research, active areas of clinical trials once they have got FDA approval. And so it's always a dynamic process. And who knows, many of you might be already engaged or would be involved moving forward in one of those trials. Okay. Now, this is, a favorite. Many of you already know. How many of you have heard about Glivec? Many of you, right? And exactly. So Glivec or imatinib is something that's extremely popular because, in multiple reasons, this was one of the most phenomenal drug that was developed to target the BCR-ABL kinase, the patients who are suffering from CML. And Brian Drucker was the director of the Knight Cancer Institute, and he was co-founder in this discovery, right? But the most important thing for us to know here, and appreciate that the way it works. So typically this is an oversimplified cartoon, right? Schematic representations showing that BCR-ABL is binding to its substrate, and it's an ATP-dependent process. And then in the presence of that, it basically phosphorylates the substrate, right? And so here is a little bit more expanded picture that in presence of its kinase domain, BCR-ABL, which gets transformed, binds to its substrate, and then the substrates gets activated through phosphorylations. And so what they have utilized, they have exploited this strategy, and they have designed the drug, discovered the drug in a way that basically intervenes in the substrate interaction pocket. That means, the Glivec or imatinib will basically slip through the crack and bind to this ATP binding pocket, therefore preventing these competitive interactions between the imatinib and the substrate, therefore leaving the substrate non-phosphorylated. As a result, if the substrate does not get phosphorylated, then the tumors cannot proliferate, and ultimately that will lead to the tumor shrinkage or remissions. So this is just an overview. You'll get more in-depth descriptions from Dr. Macfell, but this is just to tell you how a in-depth knowledge of the the structure-function relationship, the pathways that are involved in these signal transductions in the cancer biology will help you provide more in-depth understanding of therapeutic interventions. Okay? Questions? All right. So, what we are going to do next is to go over the tumor formations, which is the multi-stage process. Okay. Now, I mentioned that tumor is a multi-stage process, so it takes a long time to accumulate. Now, I'm going to take an example of two examples. The first would be to give you in the context of colon cancer, and the next would be in the skin cancer. And what you would see that they are very similar in certain aspects and distinct in some other aspects. So let's say we have a normal epithelium on the colon, and then the first gene that was discovered by Bart Vogelstein at Johns Hopkins University was the gene which is the APC gene. A mutation in this APC gene leads to hyperproliferations of the epithelium. This was one of the first tumor suppressors that was identified. Okay. Now, as you see these APC gene mutations, that drives hyperproliferation. It goes through the process of early adenoma, right? Now, then comes a second transduction mutations, which is the KRAS mutations. Mutation in the KRAS leads to the formation of the transformations into an intermediate adenoma. Now, this intermediate adenoma, which is indicated here, there is a specific mutated cells, undergoes what we call clonal expansion. That means they divide. And if you go back to our previous knowledge, this single gene with identified with multiple mutations, at least 2 mutations, now clonally expands and starts forming the bulk of the tumor. Okay. Now, once this has happened, then there is a second, the third mutation takes place. A mutation in the gene called deleted in colon cancer and located on chromosome 18q leads to the further additional new cells. The blue cells with additional mutational burden leads to the formation of the late adenomas. And at that point comes the blow, because there is a mutations in our famous P53 molecule of the year. So which is P53, which is now located on the chromosome 17p, gets mutated, and these yellow cells, which is on top, gets further activated with additional mutation burden and drives the process. So from the late adenoma to the carcinoma. And then finally, there are additional mutations, as always expected, which are distinct. And that helps in the dissemination of the cells. At this point, you see individual cells all colored different with a purpose. And the purpose is that each one of these cells cohort might have a core mutations, but they might all have individual mutations on its own, which helps them to proliferate, invade, and metastasize to the distal lymph nodes through the blood vessels. Okay, so that basically tells you that the entire stage of multi-stage process of carcinogenesis. And if you add up, typically I have listed at least 5 mutations. And then some of these mutations are called driver mutations. Why driver mutations? And this is what you will also hear if you do rotations, your clinical rotations, those of you who do in the cancer clinics. Some are driver and some are the passenger mutations. What it means is that some mutations such as the KRAS or the APC in the case of colon cancer, they drive the process that these pathways, the cells would follow in order to be ultimately lead to their fate of transformations and metastasis. Some others, like the DCC, for example, or other genetic alterations, are passengers. That means as this bus is going through the process of cellular transformation down the road into the ultimate goal to reach their destinations into the nucleus, they gather and they take up other passenger mutations, which helps and contributes in the process of cellular transformations. Okay. Now, keep that in mind, and then we will just move it to, let's say, another tissue model like skin cancer. Now, this is already, you have seen that this is a completely a longitudinal sections. That means from extreme left to a normal skin, which is shown here, to an extreme right, which is an invasive carcinoma. An invasive what? Invasive carcinoma, and that is specifically squamous cell carcinoma, because if you go back to your very first slide, which is said, what are the different types of malignant tumors? I said carcinoma, that means a malignant cells. And then if you can further define squamous cell carcinoma, so this again touch base and helps you to connect the dots. Now, what happens here is the same process. Initial stage of transformations goes through a hyperplastic skin, benign tumor, and then over a period of time in situ carcinoma and invasive carcinoma. What I did not put in here, the different mutational burden, but very similar. But what I like to mention here that you can mimic, one can mimic in a preclinical model these individual stages for drug screening, for drug validations before running any clinical trials in actual scenario. What it means that if you have a drug, or if you are working in a drug discovery company, or if you're in FDA, each one of them would be asked to do you have preclinical studies? And if you have the preclinical studies, this is how you do it, which means that you have to mimic the process that would happen in actual human patients, human subjects. And that could be mimicked by a chemical called DMBA, dimethylbenzanthracene. If you add dimethylbenzanthracene, that leads to the HRAS mutations, specifically in the codon 61, leads to the RAS activations. And that eventually, in combinations with a tumor promoter called forbolesters or TPA, will lead to protein kinase C or MAP kinase pathway activations, essentially going through and driving this process of cellular transformations. Here, as in the previous slide, we have one driver mutations. And what is the driver mutation? It is our RAS mutations. If you see, this is a commonality that you see will stand out. You have HRAS mutations here, you have a QRAS mutations there, but nevertheless, these are RAS mutations is a driver mutations, okay? So that's one important factors that to keep in mind. Question. All right. So now for our next learning objectives for you would be what I as a cancer cell need to qualify for in order to be designated as a cancer cell and not a normal cell, okay? And so these are some of those major hallmarks of cancer, right? And I will read them loud because these are important. Self-sufficiency in growth signals, insensitivity in anti-growth signals, that means how I can escape the anti-growth, growth preventive signals, evasion of apoptosis, that means how can I escape cell death, how while the people who are developing drugs are targeting will always try to induce cell death, cancer cell death, okay? How I as a cancer cell can evade apoptosis. Then limitless replicative potentials, right? We now know that how can I gather the capacity to replicate in an uncontrolled manner is one of my important characteristic feature. And then of course, how can I invade and metastasize? And of course, do each one of those things, how can I maintain a sustained angiogenesis, so the supply of the blood vessels, okay? So these are the major hallmark of cancer. And this was proposed, you see, down in 2000 by Bob Weinberg and Douglas Hanahan. Okay, many of you might have heard about Bob Weinberg. He is an MIT professor, got a lot of contributions. He was one of the first to clone and characterize RAS. And then Douglas Hanahan at that time was at UCSF, and now he moved to Basel in Switzerland. But they developed these hallmarks of cancer, and I will show you later on. In 10 years, after a decade of their discovery, they basically updated the list with other additional hallmark of cancer, okay? Now, for the sake of time, we will just discuss some of those major hallmark of cancer that I think is important for you to understand and appreciate. Now, while you're going through, keep in mind two things, or that what are the genes that are mutated in the tumorigenesis? All those genes that are involved in each one of those hallmark of cancers are actually mutated in some way or the other. It could be those involved in cell growth and proliferation. It could be in apoptosis, repair, you name it. So this is just for your FYI, okay? And I've said it earlier also that you need at least 5 or more genetic alterations here, okay? Now, continuing with our learning objective 9, what I'm going to tell you is individually we'll go over some of the critical hallmarks, starting with the apoptosis, how I as a cancer cell can resist cell death. So this is what I can do, is I am a normal cell and I can divide, and if there is a damage, the way I get repaired, and if not, I die. But then a cancer cell, on the other hand, like I said, with the first, they get hit with the first mutations, and then we know that we have multiple mutations. They basically are escaping apoptosis and leading to uncontrolled growth. So the questions that automatically comes in our mind is, how can we modulate those pathways? Again, the signal transduction pathways that are critical for controlling apoptosis. And so two things I would like you to focus on is, the first thing is there are two major pathways, extrinsic pathway, and then one is the intrinsic pathway. Extrinsic pathway, which involves the death receptors, the death signaling, which is mediated by this TRADD and FADD, these death signaling pathways, or the Fas ligand. And then intrinsic pathway is primarily mediated through the mitochondria, okay? That means mitochondrial dysfunction might lead to, and can and often lead to, ultimately activations of signaling. And what are those signaling? All of those signaling ultimately lead to activations of caspases. Which are those caspases? Essentially activation of caspase 3 in mostly, and also in some cases caspase 9. So what is the take-home message from this slide, a little bit complex slide, is that there are two major pathways of apoptosis, which I as a cancer cell would like to avoid the extrinsic pathway mediated by the death receptors, and then the next is an intrinsic pathway, which is the mitochondrial pathway. Both the extrinsic pathway or the intrinsic pathway can ultimately lead to activations of caspases. That conversion, there is a multiple stage. You don't have to remember that procaspase to caspase. If you want to, you can, but the ultimate goal is activations of caspase 3 mostly, or in cases of caspase 9, which are proteins, which basically triggers controlled cell death, which in a biological term is called apoptosis. Okay. So that means, how can I be helped? How can I be helped in order for me to escape the apoptosis, right? That means cells have to undergo some changes to facilitate me, to allow me to grow by escaping the different routes of apoptosis, right? So in such a scenario, there could be multiple scenarios, okay, to add to your complexity. So that means there could be a scenario where there could be death receptors, impaired receptor signaling, period. In some way, the signal is impaired. That means the death domain is missing, or anything that means reduced expression of the death receptors. In either ways, there is impaired receptor signaling, the first situation. The other scenario could be that overproductions of anti-apoptotic proteins or under expression of the pro-apoptotic proteins. Okay, what does that mean? So that means this is a fulcrum. So that means that fulcrum has always have to shift in this way. That means that there would be more production of either proteins that can prevent cell death, allowing me to survive, or less productions of the proteins that would promote cell death, correct? And then there are a few other options on the table. There could be mutations in the P53. Of course, in P53, you know that it drives apoptosis. The reduced expressions of the caspases. We now know that caspases are the ultimate, final destinations, activation of caspases, typically caspase 3 and caspase 9, or increased expressions of modulator of caspases like inhibitor of apoptosis, IAPs. Okay, so these are all these different possibilities how one can control apoptosis. Question. Now, the next is enabling replicative immortality. In simple word, how I as a cancer cell can keep on divide and proliferate and probably be happy lifelong. Okay, in ideal scenario, that's not the case, because a normal cell, a normal somatic cell in our body, every cell in every tissues has a limit. They undergo chromosomal, telomerase shortening. So on the two tips, the blue, tips are the telomeres. As each of the cells in our body divide, there is telomere shortening. And as the telomere shortens, ultimately this cell would die through a programmed cell death process. Now, what happens is in cancer cells, this process is mitigated. There is prevention, the apoptosis is prevented. That means these cells have a mediator to maintain the length of the telomeres. That's it. So what you would appreciate here, the major difference between the normal cell and the cancer cell here, the length of the telomeres are maintained. So obviously, I would ask, and I have a bigger interest because I'm a cancer cell, I would ask, so how can I get away by evading apoptosis and maintaining a cellular immortality status? Now, the answer to that is two. Again, the two things, there are two key players here. One is the telomerase protein, telomerase, and one is a protein complex called sheltering, providing shelter. Okay, so I would be provided, myself as a cancer cell would be provided a shelter to maintain my length of the telomeres by a protein complex called sheltering, providing me that essential shelter, the home to protect my telomeres, and an enzyme, a reverse transcriptase called telomerase, is protecting me to maintain the length of the telomeres. Okay, if you are more curious and want to know what is telomerase consists of, the telomerase consists of an RNA component, which is hTERT, and a catalytic protein hTERT. If you're even more curious, this was discovered by David Baltimore, who got a Nobel Prize for that. Okay, so that's something extremely important, who was a discoverer of reverse transcriptase. Now, sheltering and telomerase therefore would be my primary, the best friend if I'm a cancer cells. My foes would like to neutralize me by inducing telomere erosion or telomere uncapping. That means at the therapeutic point of view, I would love to, that means the cancer cells would be promoted to go to the process of, cell death if by any means you can induce telomere erosion or telomerase uncapping, and there are specific inhibitors for that. There are two-way advantage. By telomere erosion, you can involve, it's a slow pathway, it slowly induces telomere shortening and reduces proliferation. Telomere uncapping is rather rapid. That means, it can induce quick apoptosis, DNA damage response, and essentially also can lead to cell death and decreased proliferation of the cancer cells. Okay, so that's again an important feature. Question. All right. Now we are moving forward to our next objective, which is that learning objective 10, which is to know a little bit more about cancer angiogenesis. Okay. Now, I've told you by this time that cancer cannot survive without food and nutrients and oxygen. And these food and nutrients and oxygens are essentially provided by are transported by the blood vessels. You know, the transporter, like, right? And so angiogenesis is in the crust of the tumor growth and proliferations, because they feed in the food and nutrients. Now, so tumors requires angiogenesis. Now, greater than 1 to 2 millimeters sphere needs a blood supply, for sure. Now, angiogenesis does not keep up with the necrotic center. And in the past, this has aroused some confusions in many of our mind, understanding how could that happen if they require angiogenesis, why they cannot keep up with the necrotic center. And I will address that upfront, telling this in a way that means as cells undergo differentiation, which goes back to our slide 3 on the differentiation, you'd appreciate that it says that highly differentiated, well differentiated, they are benign. So that means essentially my first tendency would be as a cancer cell to divide and differentiate it and be done with that. So that means I would not be happy to support if I'm going to die and ultimately that necrotic core is not supporting my proliferation and growth. In that context, but if I want to be really an aggressive cancer cells, I would love to maintain the blood flow, get more food and nutrients, and maintain that proliferative capacity. And in that capacity, I would love to have more angiogenesis coming and feed me. Okay, does that make sense? So in that aspect, if you undergo and deep dive and saying, what are my ways, what are my friends are looking at to, undergo the therapeutic strategies, that would be the pathways are VHL, the HIF and the VEGF, the vascular endothelial growth factor axis, that is absolutely critical for the growth and proliferations and maintaining me in the angiogenic happy stage. So needless to mention, these angiogenic inhibitors are very well used and right now in the clinical trials. And so you would like to go be more inquisitive and say, well, that's good, but if I am curious to know how the mode of actions of these factors, if I have to answer to my patients, let me dive deep in and know how they work. And to understand that, let me understand how the tumors work in terms of their angiogenic promotion. So here are my tumor cells. These tumor cells are producing factors. They're producing factors that are promoting growth of other adjacent cells in the microenvironment, this heterotypic concept. They are producing macrophages, right? These the mast cells and the monocytes, the macrophages, they in turn are producing other factors which are feeding into the growth of the blood vessels. And then the tumor cells themselves are producing directly angiogenic factors in the form of VEGF, vascular endothelial growth factor, FGF, that's fibroblast growth factor, PDGF, platelet-derived growth factors, and needless to mention IL and interleukin-1 alpha beta. So these are some of those. Now the tumors are not, I as a tumor cells or a cancer cells, or I'm also producing my path to grow. How I'm doing that? I'm producing factors like matrix metalloproteinase and these proteins called cathepsin, which helps in the degradations of the extracellular matrix. That means I'm creating much more scope for me to grow and divide. Divisions to help me, the blood vessels, my friends in that being a tumor cell, I'm helping my friend blood vessels to grow and divide to provide me more source of food and nutrients. Okay. And so the next slide will give you a pictorial representation of step by step how this process of blood vessel sprouting takes place. Okay. And hang in with me actually. Okay. So this is a blood vessel, and this is a single, let's say, normal blood vessels that you see in a normal cells. And they, microscopically, if you would see that there is increased angiogenesis, there are specific markers that you would see. So how does that happen? I just mentioned to you that during this process, these proteases, this MMP9, heparinase, cathepsins, these are the ones that helps, these once produced, they helps in the cleavage of the basement membrane with the response to the angiogenic stimulus. In the response to the angiogenic stimulus, then the endothelium, which is the major cell of the blood vessels, they starts dividing and migrating. As they divide and they create a little bit of extra space, because space crunch is there, housing is a big problem, right? And so that then they start dividing with a little space and they proliferate. And these angiogenic cells, they form vascular tubes, they undergo maturations, and they basically provide the path for these blood vessels to allow this food and nutrients to, as they mature, to grow and provide more food and nutrients to the primary cell, which is the primary cancer cells. And thus providing also a path to future growth and invasion and metastasis. Okay, so that is how all of these cell types, I said co-conspirators of crime, they are all actually working hand in hand and in this side by side in order to promote and facilitate growth and interactions. And so I will take this advantage and also introduce that, well, you now know the cell biology, you know the mechanism. So then at the end, what do I get out of it? So this is what you know, that what are the factors? So VEGF is the factor, right? Vascular endothelial growth factor. It binds to the receptor, and this is everything. It's all the play. This is through VEGF receptor signaling. This is the critical pathway. VEGF receptor activates MAP kinase signaling, as I said, but it also helps integrates other signaling pathways like focal adhesion kinase, that FAK, then the PI3 kinase, and then the PLC delta signaling pathways. And so all of these signaling pathways go hand in hand, and then they merge and they converge to promote enhanced angiogenesis and providing a recipe for them to grow, and providing the clinicians and the discovery people to identify clinical intervention strategies to intervene each one of these processes. And that's what my next slide is. And so the question would be, can I deal with that? I say, of course you can deal with that. So that means you can have tyrosine kinase inhibitors, the first thing, like PDGF receptor inhibitors, VEGF receptor inhibitors, right? And they can work. You can inhibit the extracellular matrix remodeling so that the scope to grow is limited. So what you can also do, you can develop VEGF inhibitors, VEGF receptor inhibitors. Also, you can also develop inhibitors that can prevent endothelial progenitor cells recruitment here, okay? But keep in mind, so all of these are the multiple scopes. So that means it's creating a lot of jobs, creating a lot of opportunities, creating a lot of flexibility to future discoveries. But at the same time, it's kind of adding to the complexity that you can reach the same goal of inhibiting angiogenesis by targeting the different one of the several pathways in multiple ways. But this is important. Okay, question. Yeah. Yeah, there are VEGF receptor inhibitors on the market. Yeah, absolutely. There are VEGF inhibitors, they are being under clinical trials, and receptor tyrosine kinase inhibitors are there. So there are already, but there are more because each has some specific. There is cancer subtypes, and those are all complexities which we can always discuss. But probably due to limited time, we may not have time enough to go into that details. Now, so we now know angiogenesis. We now know that there is growth and proliferations of this, and we now know that blood vessels provide food and nutrients. So the question obviously would add to your mind by this time that what are the factors associated with malignancy? Okay, and so the first two major things: alterations in cell-cell interactions and formation of the new blood vessels. Why so? You will see in my next slide, because blood vessels are the major carrier of those disintegrated cells, which basically goes through the blood vessels and into the distal organs. And there is a mechanism of actions. Now, these two pictures, just for your FYI, as I say that, this is on your left is a metastatic the liver cancer, and when you make sections and then look under histology, unless and until I really put these arrows here, it is impossible to actually disseminate with which is a normal cell and which is a tumor cell. But that's a different question. That's on a different day. Now, this slide, it kind of tells you everything that I'm going to say, after this. And so what it basically tells you that we have, so when in a benign tumor, where the tumor is confined and you have limited blood flow, then everything all looks good. As there is a breakdown of the basement membrane and then the disintegration of the cells, then these capillaries, which are blood vessels and capillaries, they are the pericytes, like I said, they're increased sprouting. These capillaries are home and helps in the transportations of the disintegrated cancer cells. And through these disintegrated cancer cells, through their blood vessels, are traveling and with the help of a specific mechanism, and we'll explain that later on, adhere to the capillary wall, escape from the capillary, and then undergoes extravasation, is a process where they go and invade into distal organs called metastasis. Okay, so that's in a nutshell. Now, I think I'll stop here with this slide. So in a nutshell, what we need is to understand the whole concept of biology, invasion, and metastasis. Okay, so that means a cell has few steps to follow. They bind to the basement membrane. They induce enzymatic digestions of the basement membrane, which allows the cells to pass and slip through this basement membrane. Then they undergoes division, which helps the movement of the cells through the extracellular matrix, through the secretions of factors, and then undergo secretions of chemoattractant molecules, which allows them to slowly migrate and go into the blood vessels. Okay, let's catch this up tomorrow, and we'll stop here. All right. But if you get time, do go over the lectures, and we will follow up and wrap up tomorrow. All right. Well, thank you again. Yeah, so there was a question, it said, is RAS always a driver mutation? And the answer to that is yes. And I think I have already mentioned that. Thank you.